Hi everyone. Thank you so much, Marcy, for the introduction as well as the invitation. Hi everyone. Thank you so much for joining over Zoom, in person, and online. Um, yeah, I'm excited to be here today, and we'll just begin with the presentation once everyone's in. Okay. So the purpose of today's presentations in Tools and Tech is threefold. And I'm glad that I see most of the first and second and third year students here because this is primarily for you. So the first thing that I want this presentation to come across is to show you how you can use existing data to uncover some really interesting and insightful biological revelations. The second is the value of wet lab validation and how it sort of makes your observation more strong and gives you even more confidence. And lastly, I want this presentation to show you how you can use simple methods to uncover really interesting biology that did not existed before, just because those methods or those analyses are not in the literature right now. So to begin with, we're going to go over this presentation in three folds. I'm going to talk to you about major histocompatibility complex. It's a mouthful. It took me a while to learn itself, so I'm just going to use MHC instead. I'm going to talk to you why MHC is important in cancer. I am co-mentored by Arul Chennaiyan and Marcin Cieslak. Both of them are heavy in terms of cancer biology. Next, I'm going to talk to you about our strategy, what we have done to uncover the regulators for MHC. And lastly, I'm going to talk to you about some results that we have found, which are super interesting and how we have validated them in the wet lab. So to give you a brief of the history of the major histocompatibility complex, this is a very interesting gene molecule. The reason why this is interesting is because it's divided into two classical features, the MHC class 1 and the MHC class 2. The MHC class 1 mostly deals with those proteins which are digested into peptides inside the cell. These peptides are taken up by the MHC class 1 molecule, and this complex of MHC class 1 plus these peptides then go to the cell surface and presents itself to CD8 T cells to bring about an immune response. MHC class 1 is said to be expressed in all somatic cells. In comparison with MHC class 2, this deals with those proteins which are digested outside from the cell into peptides. They make a complex with MHC class 2 molecule, and when they present themselves on the cell surface, they attract CD4 T cells instead. Classically, MHC class 2 is supposed to be restricted to antigen-presenting cells only. This is very interesting molecules because this is perhaps the only protein associated for 4 to 5 Nobel Prizes. It's highly polymorphic, which means that it's just highly variable and very hard to study. And it is associated with many diseases, including cancer, and that's why we're interested in them. So the MHC class 1 and class 2 can be mutated both at the genomic level and can also have mutation in terms of losses in the transcriptomics level. So MHC class 1 and class 2 losses were studied by Brody, who was also a graduate from our lab. He studied this in a number of different cancer types. We also have this review paper, which shows MHC proteomic loss across different cancer types. So you can see that MHC is lost in most cases, especially you can see prostate over here. FRAD is lost in more than 50% of the cases. So if it's a transcriptomic loss, maybe we can bring it back. Let's try to understand MHC class 1 and class 2 more so that we can understand the rest of the presentation. So MHC class 1 is expressed in all somatic cells or all nucleated cells. It presents antigen from the intracellular pathogens, and then those antigens are then presented on the cell surface. These are endogenous proteins, and they interact with CD8 T cells, and they act as surveillance by detecting and eliminating these cells by the immune system. MHC class 2, however, is supposed to be expressed in antigen-presenting cells. These are those proteins which are first engulfed by the cell and digested in the antigen presenting cells, which are called the APCs. So they are basically dealing with exogenous proteins and they interact with CD4 T cells. So very different features, yet very similar sort of genes, right? So they're both MHC, but they're both different classes of MHC, and they both have different interactions with the immune system and how they basically present themselves. So given that their importance is so high and there are 4 to 5 Nobel Prizes with this gene, they have been studied in a huge amount in literature. However, according to this review paper itself, the mechanism behind the basal expression of MHC is still very limited. And the reason behind that is that most of the data that we have so far is being confounded by cell type heterogeneity. So to identify the mechanism of MHC transcriptional alteration across different cancer types, we need to understand how cancer cells evade the immune system. So we need to go on a more cell type specific analysis. And one of the ways to do that is to use single cell data. But before we go into that, this is what the literature currently has with regards to MHC regulation. So MHC so what we have learned so far is that MHC is a very important gene with regards to cancer. It's lost in majority of the cancer, but we still don't know why that loss occurs. So we already know that MHC is regulated by this enhancosome, which is NLRC5, C2DA, RNFY, etc. genes, transcription factors, but they're still unknown regulators that we are not aware of that regulate the MHC class 1 expression. And think about it, if we can understand these regulators in the basal or normal setting, we can then understand which regulator is dysregulated in cancers and in which types of cancers. So what we already know is that MHC expressed in all somatic cells, MHC class 2 can be expressed in epithelial cells under inflammation. MHC class 1 and class 2 reside in the same genomic locus, but they are regulated very differently. NLRC5, C2TA are the master regulators of MHC class 1 and class 2, and MHC is repressed in many cancers. So we already know this information. What we still do not know is that for MHC class 1, how variable is the MHC1 expression. So we know that it is expressed in all somatic cells, but is it expressed in the same level? Is it a housekeeping gene? Does it have some variability? We don't know that yet. So what cancer types loses MHC1 or upregulates MHC class 2? So cancer is a weird phenomena. So it has a very different reaction to different things. So we still don't know a lot of things about MHC. Can we observe MHC class 1 repression at the mRNA level as well? And lastly, what are the differential regulators of MHC class 1? Like I showed you before, we still don't know what all the major players are. For MHC class 2, can MHC class 2 be expressed in the baseline normal epithelial cells? Literature or your course books will say that MHC class 2 cannot be expressed in antigen-presenting cells. However, with the current literature and what we have, that's not entirely true. There are cases in which MHC class 2 can also be expressed in epithelial normal cells. Next, the question is, what are the differential regulators for MHC class 2? How does MHC class 2 expression by cancer cells affect the immune system? How heterogeneous is the expression between malignant and epithelial cells? What are the pathway differences? How does MHC expression link to clinical outcomes? So these are the questions that we had once we went to this project. So this is where we wanted the computational biology analysis to fit in. So in order for us to answer all of these questions, what we did was a landscape study of 0.73 million cells to understand MHC regulation. The way we have broken down this study is into 3 different cases. So we don't know how MHC is expressed in the normal setting. So let's try to gather all the normal tissues. 18 different tissues, 124 epithelial cell types. We don't know which cancer react how with MHC class 1 and class 2 expression. So let's gather all the cancer-related data sets. 43 different data sets, 20 cancer types, around 500 patients. We don't know how MHC can be affected by immunotherapy. So let's gather all the immunotherapy-related data sets. We have 174 samples, 8 different data sets, 9 different cancer types. So a large compendium of more than 50 pan-tissue, pan-cancer, single-cell RNA-seq data sets that we collated throughout this study to understand MHC regulation. So very simple question, how is MHC regulated using existing data? So this is how our study is manufactured. So the first step of our study is just to see in which epithelial cell type is MHC class 1 and class 2 expressed, what's the level of their expression. The second part of our study is to understand the regulators. What are the differential regulators for MHC class 1 and class 2? Can we validate that in the wet lab using some form of CRISPR screens? The third part of the study is that let's go into cancer now. How is MHC class 1 and class 2 expression different across different cancer types? The fourth is, are there any interesting cancers that we can focus on? We said, yes, this is prostate cancer, super interesting with MHC class 1 repression. The fifth part of the study is that, are there other interesting cancers that we can focus on? There are. Colon cancer actually upregulates its MHC class 2 expression in the tumor cells. And lastly, what are the clinical associations? So we have the study plan on the left-hand side, and then we have our experimental validations on the right-hand side, because your bioinformatic validations can only be true once you have validated them in the wet labs. So in order for us to understand MHC class 1 and class 2 expression in the normal setting, we thought about using this tabular sapiens data set that was at that time recently published. It had 18 different tissue types, 124 epithelial cell types. Our approach was use this data, make some algorithm to nominate transcription factors, and then see which transcription factor is affected in different cancer types. So this pyramid scheme of exploratory analysis, mathematical modeling, and then finally some experimental validation. Once we started analyzing this data, we realized that the data is not perfect. People have not done best jobs in depositing their data, and always question whatever data that you have. So the problem that we had this data was, it was not granular. So for example, some of these tissues had only one cell type. The second was missed annotations. So they had hepatocytes in the heart. That cannot be possible. Hepatocytes are liver epithelial cells, and somebody also tweeted about this. And lastly, their immune cells also existed in the epithelial compartment. So once we were able to identify these problems, we said, like, okay, now we need to actually re-annotate, resort, remove all of this pipeline again in order to get some really pure data out of this. So what we did was we downloaded our 18 different tissues. At that time, it had 79 epithelial cell types. We separated each compartment into individual tissues, reclustered them, removed the non-epithelial cells, manually relabeled them, and then we ended up with 24 epithelial cell types and 18 different tissues that we believe or trust in our downstream analysis, because everything is dependent on how it flows. So the first question, yes, sir.
So after you did all this cleaning up with the data, you had many more cell types.
Yes.
You might want to tell us a little about that.
Of course.
And not very, even just 10% fewer cells. So that was pretty efficient in finding additional cell types.
Yes. So thank you for that question. So some of the cell types are actually rare, so they're hard to identify. Tabular sapiens used an automatic cell type labeling tool in which they ran this tool and it automatically gave you, okay, this might be this cell type or this might be this cell type. They did not go in detail about how to annotate some cell types which are rare. They might be in small in numbers, but they exist. So that's where the manual annotations are super needed, because you have already these databases which have cell type markers. For example, ionocytes exist in the lung, but they exist in very small quantity. Pennant cells exist in the epithelial of the small intestine, but they're very small quantity. So in order for you to identify these cell types, you have to go to these databases with cell type markers and then look for them in order for you to see them. Does that answer your question?
You did all that yourself?
Yes.
Impressive.
It took us some time, but we did this. This was needed because we needed a good normal single cell RNA-seq data set in order for us to identify regulators, and it paid off. Let me show you how. So the first thing that really pops up, even if I like just show you this data, is lung, right? So lung has high expression of MHC class 2 and class 1, and this is only epithelial cells. So, but however, this is not novel because lung is known to express MHC class 2 in their epithelial cells, especially AT2 cells. So this is one thing that we noticed, which is a good validation that our analysis is working. The second thing that we notice is that there is a variability in MHC class 1 expression across these 18 tissues. So, which basically suggests that MHC is not housekeeping. There's a variability. Why that variability exists, we don't know yet, but it's definitely not the same level across different tissues. So, but let's go into more details because we're not interested in just tissue level. We're interested in the epithelial cell type level. So you can see over here, the blue is MHC class 1 and the yellow is MHC class 2. First thing, again, eightfold difference between all of these 124 epithelial cell types for MHC class 1 expression. And then you have some cell types which have very high MHC class 1 expression, some cell types which are very low. SNR cells are known to express MHC in a low quantity. This was shown in a thesis very long time ago. Nobody followed up on this. But MHC class 2, you see 82 cells in the lung, goblet cells in the trachea, goblet cells in the lung, club cells. All of these cells are interesting because they express MHC class 2. And then there are some cells which have almost no expression. And this is what literature says, that it should not have high expression of MHC class 2. But we're not done yet. So we want to understand the rare cell types, epithelial cell types that express MHC class 2. So those rare epithelial cell types are, for example, the prostate. So the normal prostate epithelial club cells have some expression of MHC class 2, right? So this is some expression. I'm not saying this is high. The high is 82 cells, but some have expression of MHC class 2. And then we also nominated other epithelial cell types that have some expression of MHC class 2 that is not known before. But this is a bioinformatic analysis. There might be external factors associated with this. For example, once I showed this data, Dr. Matthias, he told me that tabular sapiens patients were brain dead. So this might contribute to the MHC class 2 expression in the lung because there are tubes going down and things like that. So you always need to have some sort of validation either in your lab or through some external database. So we fortunately have a really nice lab and people who are willing to do this for us. So what you see over here is a normal prostate tissue, and the brown expression that you see over here is the MHC class 2. So we were able to get some staining or enrichment of MHC class 2 in a normal prostate epithelial cell types. And if we look at this in comparison, so this is a conventional looking prostate gland with minimum MHC class 2 expression, but then these are these atrophic prostate duct with increased MHC class 2 enrichment. So there is some biology that suggests that MHC class 2 can be expressed in the prostate tissue in a normal setting, not even in inflammation. So we wanted to dig this more deeper. Dr. Rahul had already done this PIGR staining previously in one of his papers. He saw that this trend, this morphology resembled a PIGR morphology. Interestingly, PIGR is also a marker for club cells. So we decided that let's look at another single cell normal data set and validate this observation over there as well. So over here, they have already Hillock identified, this is a Henry et al. Data set. They have already identified club cells. This is the expression of the club cells. And then we have PIGR. And we have MHC class 2 coming from these normal club cells. So this suggests that MHC class 2 can be expressed in a normal club prostate cell type. And we know this is club because PIGR is a known marker of club cells. We also did some co-expression of PIGR MHC class 2, and we saw that in a rare population of prostate epithelial cells, we have co-expression. So this is something which is not known before. We observed this in a normal setting and then validated that in the wet lab setting. And then we had some additional validation. I'm not going to go into all of them, but all of them except one of our single cell observations was validated in the wet lab setting. So now I am coming towards the more important part of this presentation. So our objective was to see where MHC class 1 and class 2 is expressed, answer some questions related to that biology, then come into the question of why that's availability, why is MHC class 1 and class 2 expressed, how is it expressed, what are the differential regulators. If we can identify those differential regulators in the normal setting, are they dysregulated in cancer? So in order for us to answer the first question, what are the regulators of MHC class 1 and class 2, we went back to this diagram again. Okay, so we already know because of the huge history that MHC has that there are some major regulators, NLRC5, C2TA, and things like that, that are known to regulate MHC. So how do we understand the additional regulators? Let's go to literature. So there are a lot of tools and methods already published in order to understand transcription factors in a single cell data. Almost all of us who work with single cell data know about CNIC. There's BitFam, there's Dorothea. Then there are additional papers or methods that I didn't know about, which is Triangulate, DeepSeam, Inferlator, FigR. There's so many. So we did not wanted to make a new method or make a new approach to uncover the regulators of MHC. However, once we started running these tools and using these tools in our analysis and then reading the papers, we realized that these tools are mainly focused on identifying transcription factors in order to identify the cell types. Right. So for example, CNIC argues that transcription factors are the reason why you can identify a particular cell type. They basically govern the morphology or the expression or the identity of a cell type. And that's what CNIC is arguing, that if you can identify the transcription factors, you can identify the cell type. We're not interested in identifying the cell type. Then we know that MHC is a highly polymorphic gene, which means that we cannot use single cell ATAC-seq because the promoter of MHC is so variable. So there's a lot of constraints that we had in order for us to answer our question of what are the regulators of MHC. So we said that instead of doing this and not coming to a conclusion, concrete conclusion, let's just go back to basic mathematical analysis. So this is our question. So let's just answer this question by using simple correlations, right? So we want to correlate MHC class 1 expression in individual cell or cell type against all the known transcription factors. So the transcription factor with the highest correlation can be a factor responsible for MHC class 1 expression. I'm not saying here that correlation is equals to causation, but let's argue for a second that this could work. So what we did was that we made this pipeline, a very simple pipeline to follow. So you already know about univariate regression models, which means that Spearman correlation, Pearson correlation, simple regression models. And then we have lasso regression, which is more like a multivariate regression models. So what we did, and it's a little hard to follow, but I'll try to make the best out of this. We made this a pipeline, which is like a data discovery master regulator pipeline. It's not fancy. It's easier to follow, but I'll try to explain this. Good. Okay. So the first thing that we did was that we used the single cell expression data. So this is your single cell expression matrix. You have MHC class 1 and class 2, depending on what you want to study. And then you have all your 1,300 transcription factors, right? And then you have cells on the top, genes on the row, okay? So this is your matrix. The first thing that you do is that you split the matrix into individual cell types. So for example, prostate, luminar, 1 cell. And then you have lung, 82 cells, right? And so on, all the way for the 124 epithelial cell types. Then once you have this, you compute the correlation, the Spearman correlation between MHC class 1 and each individual epithelial cell type. So you will result in all the transcriptions, like 1,300 transcription factors. There are 2 scores or their correlation coefficient score, and in which cell type this score was computed. Okay, So you're basically running a specific Spearman correlation in each individual cell type separately to understand the best regulators for MHC class 1. Once you have this matrix, then you can scale your correlation coefficient in each cell type and then take average across the cell type. This will allow you to nominate the best transcription factors for MHC class 1 across all the epithelial cell types. You might notice that this already has a lot of flaws, right? So you're assuming correlation is causation, and you're scaling, you're averaging, you're adding all of these modifications in the method. So we're not going to use just this. We're going to augment this with something else. So there's another concept called meta cells. So if you have an epithelial cell types, for example, luminal prostate cell, it has 500 cells within this epithelial cell type. You can then use this meta cell approach to make subclusters within a cell type. So instead of having 500 cells in one cell type, you'll have 5 different meta clusters in one cell type, each meta cluster with 100 cells. So this is what we use in order to get an average or tackle the issue of sparsity in single cell, right? So sparsity is also an issue in single cell data. Your data it has a lot of zeros. In order for us to handle that, we use meta cell approach. So let's walk through this. You have your expression matrix again. You have your expression matrix split into epithelial cell types. Each epithelial cell type has a separate matrix. Then you use your meta cell approach in order to sort of congest your matrix, get some averages instead of the actual values so you can handle your sparsity. So once you have this, you compute the meta cell matrix for each cell type. Then you do the same thing that you did before. You correlate MHC class 1 or class 2 expression in against each transcription factor in each cell type. So you're left with this matrix with the transcription factors, with the correlation coefficient, and within each cell type. Lastly, to get the best transcription factors, you scale within the cell type, and then you take average. But we're still not done yet. We still don't believe the data, right? Because again, there's a lot of assumptions that goes in. Univariate regression models have a lot of issues. You're assuming correlation is causation. So we're not happy yet with our analysis. So let's add some feature selections into this. So the first thing that we do with both of these matrices, this one from single cell and this is one from meta cell, we combine this using averages. So just average the correlation coefficients between these two matrices and leave it aside. Then in a completely new scenario, you use lasso. Lasso is a way to nominate features, and it handles sparsity much better than simple correlation. And it can nominate those transcription factors or those features that would be the best across all features. So we perform lasso to nominate the transcription factors and in more than 0 cell types. So if this transcription factor had a value or was nominated by lasso in more than 0 cell types, then we overlay it with the correlation results that we got previously. So this is where a little of the intuition comes in. So you're putting some thresholds, you're using your own brain. You don't really have a perfect way of finalizing which transcription should go in your model or which should be the finalized model, but you're using some threshold in order. And the way you use this threshold is that you look at the entire list, you see the known transcription factors, and sort of like figure out where should I put that threshold. That's the first thing that we did. The second thing that we did is that we used some RMSE scores, a root mean square estimations, to figure out what should be the best threshold. But in any case, out of these 1,332 transcription factors, using our average scores, top transcription factors based on this average, and the top transcription factors based on lasso, and the bottom, because we also wanted to have some negative regulators based on lasso, we were left with around 124 transcription factors. But we're still not done yet. So we got this result from Spearman correlation, lasso, so both univariate and multivariate regression, meta cells and single cell. We got this, so this is a previous number. We got 104 transcription factors. We're not done yet because we have other tools, right? We have so many other tools for feature selection, for nominating the best genes. Let's try to incorporate that as well. So we used Genie3, Baruta, and Lasso again. We use all of these three regression modeling tools to nominate the best transcription factors from these tools as well, and then figure out whether they overlay with our previous analysis. And the ones which overlaid, we decided that these 35 out of the 1,332 transcription factors are the best ones to predict MHC class 1 expression in a normal setting. And this is the downstream analysis that we hope to do. But this is how we came to our transcription factors for MHC class 1, and we did the same thing for MHC class 2. This is a normal setting across multiple different tissues and epithelial cell types. But we're still not satisfied, right? So this is a bioinformatic analysis. We need to validate this in the wet lab. We got these results, but does it actually predict, right? So I don't have much time, but this is a more generalistic view of how the pipeline looks like. So before going to the CRISPR screens or validating in the wet lab, let's validate this in the bioinformatics setting itself. We made a regress pipeline. We got these transcription factors. The first question is, does these transcription factors also have the known transcription factors for MHC? If we did this regress pipeline, we should get NLRC5, we should get C2TA, NFY, and all those transcription factors that we know exist based on literature. So our enrichment analysis showed that we do have a strong enrichment, at least p-value is significant in terms of reported. So 18 transcription factors that were previously reported to be regulating MHC class 1 existed in our 104 selected transcription factors as well. So this basically gave us confidence that our pipeline is working in nominating MHC class 1 molecules. The other thing that we did was just to make sure, this is one thing that I explained already, but we used Genie3, Lasso, and Baruta to validate this. This is how we justify the 35 transcription factors we use in RMSE score. This is where it plateaued, and we said that this might be the best number of transcription factors to include. So once we had this, the next question is, do they perform better than reported transcription factors? What if we use the existing transcription factors list and use it to predict the MHC? If that is better than our approach, what value are we adding, right? So then the second question is, are they performing better than randomly selected transcription factors? What if we put some randomly transcription factors in our model and they can still predict MHC better than our model? And the last question is that we just use tabular sapiens for this. Can this be true with other data sets as well? So we went through these questions one by one. In terms of reported, so you can see that these are the reported transcription factors. These are our nominated ones. And this is when we have reported and are nominated. And lower RMSE suggests that it's a better model. So lower RMSE, higher R square suggests that it's a better model than just reported. And this is true for the individual cell types all clumped together, and it's in, in separate cell types as well. The second validation was that, okay, so reported transcription factors are like, what, 18? We have a list of 104 or even 35. We have more transcription factors. Maybe just having more transcription factors makes the model better. So what we did was that we randomly selected the same amount of transcription factors that we were putting in the model. So, for example, if our model has 57 transcription factors, we randomly selected 57 transcription factors, made bins out of it throughout the list of 1,300 transcription factors, removed our transcription factors from that list, made 500 different models randomly generating those transcription factors, and then see whether our model can perform better as compared to a randomly selected set. And for both of these graphs shows the same thing, that our model was predicting a better R square as compared to those which are randomly selected transcription factors. So just to make sure that the number of transcription factors are not affecting the model itself. And lastly, we also validated this in a non-tabular sapiens data set. So our reported plus nominated list has a much higher R square compared to random selected transcription factors. So this is our putative network in terms of MHC class 1 and class 2 regulation that we have done, that we have collated based on our bioinformatic analysis. But since we're not done, we still have a wet lab validation to go further into. So we have a nice lab. So the immuno team at MCTP, which is the Michigan Center for Translation Pathology, YIBAO, did a targeted CRISPR screen for us. So MHC class 1 CRISPR screens have already been done in literature at least 5 different times. But what they have done is that they have did this CRISPR screen across all genes and then nominated their transcription factors or their genes. We have a much targeted approach. In terms of the CRISPR screen, so I'm not going to go into the method of how we did this, but basically you do some normalization, filtering, calculating fold change, do some linear normalization, and then you get your gene level estimation. It's not perfect. So there are things that you can manipulate in terms of CRISPR analysis. But because of time, I'm not going to go into the CRISPR analysis. I'm just going to show you the data. So based on our testable targeted CRISPR screen, we were able to validate some transcription factors and genes that are known to be positive regulators of MHC. So we have STAT1, which is a regulator for MHC, CREB1, regulator for MHC, and LRC5, interferon-1. This means that our screen is working. So the red basically shows that the CRISPR screen is working, and everything else is sort of novel that we nominated. And in the negative regulator category, we know that SUS12, SUZ12, and EED is a known negative regulator of MHC class 1, and we're getting it in our CRISPR screen, which means again that the CRISPR screen is working, and everything else other than the red are sort of novel regulators. So we have not done this CRISPR screen in just one cell line. We've used 3 different cell lines. I'm just showing you the data for one of them. And because of 2 additional cell lines, we have much more robust and results now. But basically, this is showing you that the bioinformatic analysis, the ones that predicted the MHC class 1 regulators, are also being validated in the CRISPR analysis. So I have 10 minutes. I will try to go through this as well. So let's go back to the objective. The objective was look at MHC class 1 and class 2 expression across epithelial cells, across different tissues in the normal setting. Then nominate the transcription factors of MHC class 1 and class 2 in these normal tissues, right? And the actual objective of all of this was to figure out whether they are dysregulated in cancer, right? So previously, all that we talked about was the normal data. Now we're moving into cancer. So in order for us to answer what happens in cancer to MHC class 1 and class 2 and the regulators, we made this large compendium of single-cell RNA-seq data sets across these 20 different cancer types, patients, malignant cells, as well as epithelial cells, using different databases and inclusion criteria where we're only using 10x primary tissues with malignant cells, with raw counts, and at least 20 cells per patient. Okay, so this is an argument of why we need single cell data. If we just look at the bulk TCJ data, we don't see a difference between MHC1 and class MHC2 between tumor and normal, which means that bulk is highly contaminated. We cannot answer our questions using bulk data. In comparison to single cell, we have a much better repression seen in malignant cells for MHC class 1 and class 2, both by paired and unpaired analysis. So this gives us a confidence that we need to use single cell data for this. So this is when we have an interesting analysis because we see the cancer types on the x-axis and the expression of MHC class 1 and class 2 on the y-axis. So the interesting cancer types are perhaps the kidney cancer. So kidney cancer has high MHC class 1 and class 2 expression. So what you see in this plot is the fold change between malignant and epithelial cells for MHC class 1 and class 2, and each bar is one patient. So you have a higher fold change for MHC in the CCRCC, which is the kidney cancer, and a much lower repression of MHC class 1 in the prostate. So the kidney cancer has high MHC class 1 and class 2, and the prostate has low MHC class 1 and class 2. And this is how we nominated which interesting cancer types we can look into. So whenever you have some interesting insights from bioinformatic analysis, you need to do some wet lab experiments. So from our wet lab team, we were able to confirm that in kidney cancer, which is here. So in kidney cancer, you see both expression of MHC class 1 and class 2 significantly over here. In prostate, in primary, you see some expression for MHC class 1, maybe some expression from MHC class 2, but no expression from the metastasis of the prostate. So this basically confirmed our observation of where MHC class 1 and class 2 is expressed. So one of the things that I wanted you guys to to share with you is that if you have done like a huge amount of analysis for your research, looked at different databases and so on, you can always like make a review paper out of it. So this is our review paper detailing the single cell data set that exists out there, making some groupings about which, for example, this is primary data, this is from aggregated data, you have different cancer types as well. So basically you can actually you can actually get something out of it as well if you have done a lot of literature review. So I was going to show you where this data is located, but I think you can just go to the review paper itself. Since we are short on time, I'm not going to show you the colon cancer results, but just know that MHC class 2 was high in MMRD patients. MMRD patients are mismatch repair deficient. They have higher tumor mutation burden and more likely to respond to immune checkpoint therapy. And this is why we went into immune checkpoint therapy for our last analysis. But for the immune checkpoint therapy in the last couple of minutes, I wanted to show you that we compiled a couple of single-cell RNA-seq data sets that had immune information. So immune therapy is basically when you try to use your body's own immune system, try to upregulate, activate it in order to fight cancer. We see that MHC class 1 and class 2 were high in those patients that were favorable or had a good response to immune checkpoint therapy. Once we had this data compiled again, so this is an avenue for you to use as well. If you have compiled large data sets together that did not existed before in a harmonized way, you can publish that as a data descriptor. So we recently published this compendium of 9 different cancer types, 174 patients of all the immune checkpoint therapy data sets that existed in terms of single cell. And we were able to publish this in Nature Scientific Data. So since you are early years, think about this, like how can you use your information? How can you use your previous analysis, the work that you have put in to get something out of it? And this is just some of our results in terms of how MHC class 1 and class 2 scores. So now you have signatures, right? So you're able to identify transcription factors that predict MHC class 1 and class 2. You can turn them into signatures and see whether you can find a response in terms of Kaplan-Meier plots or overall survival of your patient treated with cancer, treated with immunotherapy, or with the regular therapy. So immunotherapy, you can use this ICB data set that we compiled. For regular therapy, you can also use like TCGA in order to understand this. So we have some ongoing experiments that are currently in the process. And just a summary, this is a first large-scale study to interrogate the regulators of MHC at the single-cell level. We have developed a data-driven approach to identify universal and cancer tissue-specific transcription factors associated with MHC. This study has resulted in a testable hypothesis, and the CRISPR screen is currently under development. We have some really novel observations that we have so far, and we have some future validations currently in the process. And for the terms of acknowledgement, the most important slide of the presentation, I am again co-mentored by Dr. Martin Cieslak and Aral Chanayan, and we have a huge lab and a lot of people to help us out with our experiments and our bioinformatic analysis. And thank you. Yes.
Excellent presentation. Hi. So I had a few questions about the first correlations that you're performing. So with single cell, there's a high dropout rate. Do you just use the positive expressing cells, or do you somehow like normalize to increase the values of 0? How do you?
That's a very, very good question. So we, in our analysis, wanted to nominate both positive and negative regulators. How we tried to use imputation for it, like magic. We realized with magic, it introduced like undue correlations that are not supposed to exist. So what we ended up doing is that we said that we're only going to nominate the positive regulators for MHC class 1 and class 2. It's very hard to get negative regulation or negative correlation in single cell data because of your points. Yeah.
Interesting. Thank you. The other thing was because when you generate the correlation coefficients, they're usually between 0 and 1. So do you need to scale them, or do you just average them up? So why did you have the scaling and averaging?
That's a good question. So we needed to scale them because in different cells, you have different cell types, you have different number of cells as well, right? So that might just introduce some variability in the data that we do not want to handle, some technical issues. That's why we have to scale them within each cell type and then take average of them. But we debated about that like a couple of weeks, and we did both ways around, and that's when we arrived at this. So yeah, whenever you have these sort of questions, there's no one answer. You have to try all of them and then see what works best. I mean, that's a good question.
Thank you.
Yes. Hi.
Yeah, great talk. Same thing like pipeline, I guess. I know you looked at the like TF code expression sort of after the all the correlation and the like lasso modeling, like step 3, part 3 right there. Wondering if you looked at TF code expression before the expression model. So I know like linearity sometimes like causing problems.
So your question is that why didn't we use lasso before we did experiment?
More so like you guys just look at the full expression of the transcription factors themselves before.
Oh, yes, that's a very, very good question. Yes, we did make those heat maps and try to see whether the transcription factors were just very much correlated together. What we realized from that analysis was that we cannot disentangle it because some transcription factors, for example, we could also see that from our CRISPR analysis that, for example, the known regulome of MHC class 1, the CREB and the NFY and RFX, these are like same family, and they have like huge correlation similarity as well. But because we're trying to discover new biology, we could not like disentangle that information, and that might influence why some of our nominated transcription factors are not working in the CRISPR screen. But that's a good question. Yes, hi.
Really nice. Transcription factors are known to be relatively low expressing for their impact. So I'm curious on how, if you had a bias in terms of expression, like were more of your nominated ones of the higher expressing transcription factors? Then maybe as a follow-up, are you planning on extending this to all genes beyond transcription factors, and what would you expect to find?
That's a very good question again. Also brought up by my thesis committee. So the idea is that we looked at this in a couple of different ways. We also looked at the correlation between, the number of cells and the transcription factors correlation coefficient that we got. We also looked at, what's the Gini coefficient for that. We also looked at, so the thing that you suggested as well. So to answer your question, we did try to understand the variability and how it can influence the correlation coefficient. And from what I remember, it was a very long time ago, we realized that in most cases, it did not. The only case where it was effective was when we have very low number of cells in a cell type. So that's when we decided we need to scale them, we need to average them. We cannot just nominate it. Because our first approach was that we want to nominate the transcription factors per cell type. So each cell type would have its own transcription factor. That would make more sense. But because of this, because of very low expression of transcription factors themselves in single cell data, we couldn't do that. That's why we had to go for meta cells to try to get some average out of it. But for example, STAT1, which regulates a lot of things, is showing up in our analysis, but it also regulates MHC. So it's hard to disentangle that. And to answer your second question, which was
beyond transcription factors.
Yes. So I tried to do this is more a generalistic view of how this pipeline would look for in any gene of interest instead of MHC class 1 and class 2. However, when I pitched this to my professors, they had very good points that, for example, we're not looking at ATAC-seq over here. Whenever we want to nominate regulators, we need to look at single cell ATAC-seq. We are not incorporating ATAC-seq because MHC locus itself is very variable and this would not help them. So in any other genes, we would incorporate ATAC-seq data, single cell ATAC-seq data. So that's where this pipeline is lacking. The other way this pipeline is lacking is that our CRISPR strain is all for MHC class 1. Right? If we want to show this method as something that is effective for all genes, we need to do CRISPR screens for like multiple different genes as well in order for us to like validate that observation. But I'm still not done convincing my professors. So I am thinking about, maybe putting this out as a separate method that can be used for all genes, but it will need to be modified a little, especially with the single cell ATAC-seq. We need to incorporate that in some way. Okay. Are there any more questions? Love to have the feedback as well. So we've done this analysis for the last 3 years. It takes time. If you're first year, second year, this is the advice for you. It takes time to learn anything new and do something new. So if you're struggling, just keep at it. You'll eventually get there. Yeah, I'm still struggling, so. Okay, thank you.